This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. In 1967, an ambitious breeding program created a new, super variety of potato destined for chips production, the Lenape. The variety was resistant to common potato diseases, but also contained high starch and low sugar content, which made it particularly suitable for the production of nicely, bright-colored chips, contrary to potatoes with high sugar content, which darken when fried. But almost as soon as the Lenape potato started being cultivated in the US, farmers complained of suffering from various illnesses after eating them, from digestive troubles to cardiac dysrhythmia, headache, dizziness, itching, and inflammation and pain in the joints. All these symptoms were pointing towards an intoxication. So after a detailed analysis of the chemical compounds, it was discovered that the Lenape potatoes have up to 5 times more solanine than other potato varieties. Solanine is an aromatic compound present in all potatoes, especially when they get green, but in such small amounts that it does not affect the human body. Back in the 1960s, measuring the solanine content in potatoes was not a standard practice in breeding programs, so this newly bred trait could not be detected. The Lenape potato was quickly retrieved from the market, and new legislation was reinforced, making the evaluation of solanine content mandatory when breeding new varieties of potato. In the 1990s, a new white potato hybrid resulting from the cross of domesticated potato and the wild potato without tubers was created in Finland. The new hybrid was resistant to various diseases, including blight and the common potato scab. Having learned from the Lenape experience, a detailed chemical analysis was made on the 2 potato species and their hybrid offspring. Interestingly, the cultivated potato showed, among other things, relatively high levels of tomatidine, and the wild potato had high levels of solanidine. But their hybrid offspring had only small amounts of tomatidine and solanidine, and very high concentrations of demicidine, another aromatic compound that was found in neither of the parents and which is quite toxic for humans. Both of these examples show how breeding can produce crops with undesired traits. In the case of the Lenape potato, there was an unintentional selection for a trait that was not measured. One of the original potato varieties that had the blight resistance also had a gene coding for a particularly efficient enzyme for the synthesis of solanine. Because this variety was not used for food, breeders were not aware of its high solanine content. When this variety was crossed with a starchy potato, the resulting hybrid inherited both the blight resistance and the solanine gene. Then followed a series of back crosses, and at every back cross, the potatoes with blight resistance and high starch content were selected and used for the next generation of back crosses. Unfortunately, not being aware of the solanine issue, breeders unwillingly also managed to transfer the solanine genes into the potatoes. As solanine content is not directly visible and was not measured throughout the breeding program, no breeding efforts were made to eliminate the trait from the hybrids, and so it was spreading from one generation to the next. In the case of the white hybrid potato in Finland, combining the 2 potato species resulted in the creation of new epistatic interactions between different genes, resulting in a new biochemical pathway for the synthesis of demicidine. The domestic potato has a hydrogenase enzyme that uses steinamine as substrate and transforms it into tomatidine. The wild potato has another enzyme which transforms steinamine into solanidine. The hybrid of these 2 species has both enzymes, and the wild-type enzyme is more reactive with steinamine, so it first transforms it into solanidine, which is then taken up by the hydrogenase of the domesticated potato and being transformed into demicidine. As demicidine is not found in neither domesticated nor wild potatoes, the breeders did not undertake any steps to eliminate genotypes that were producing it, because you simply cannot eradicate what you don't know is there. Another cause of breeding for undesired traits is when the selected trait is regulated by a pleiotropic gene. This was the case of a breeding program that aimed to improve agronomical traits in tomatoes. Ripe tomatoes are red. However, land-raised tomatoes and wild tomatoes are not uniformly red. They first gain the color at the bottom, and then it gradually spreads towards the top of the fruit. A ripe tomato can thus still appear green-yellowish on the top. This is an issue for tomato picking, because to check for their ripeness, a picker needs to turn the fruit over. It might not seem like a lot of effort when you grow tomatoes in your garden, but it certainly adds up to a lot if you're farming tomatoes for big distributions. The color pattern of the tomato is determined among others by the gene SIGLK2. This gene is involved in chloroplast production. The expression of the gene is such that the concentration of chloroplasts is high at the bottom of the fruit and low at the top of the fruit, resulting in a red to green coloration gradient after ripening. Using mutagenesis, breeders produced a tomato variety that has reduced expression of the gene in the tomato fruit. This resulted in an overall low concentration of chlorophyll in the tomato and the nice homogeneous red color when the fruit is ripe. Unfortunately, these mutant tomatoes also have low sugar content, no aromatic compounds, and no flavor. This is because SIGLK2 is a pleiotropic gene. It is responsible for the expression of more than one phenotypic trait. The chloroplasts that the gene produces not only give the green color of the fruit, they also do photosynthesis, and the sugars produced by photosynthesis are then used in various biochemical pathways to produce organic compounds responsible for the flavor and scent of the tomatoes. The knocking out of this one gene has consequences for multiple phenotypic traits that were not accounted for in the breeding program. Nevertheless, red, flavorless tomatoes are the consumer standard nowadays, as the goal of the breeding program was not to satisfy the consumers, but the producers and distributors of the tomatoes. In all these examples of conventional breeding, inadvertent selection of undesired traits is the result of manipulating the whole genome of the plant. To eliminate undesired traits, all individuals that have them need to be eliminated through the mass selection process. This is a complicated task. For its successful completion, it requires not only that the exact desired genotype is produced with random recombination and assortment of the genes in hybrid offspring, but also for the breeder to have an oversight of all plant traits, from morphology to the finest metabolical processes, which is a very complicated task. Genetic engineering techniques reduce the risk of selecting or introducing undesired traits in the crops, because these techniques only modify one gene at a time instead of manipulating the whole plant genome. But genetically engineered crops present other risks of unwanted modifications. With the classical methods, the gene construct is inserted at a random site in the genome. If this site happens to be a coding sequence, the insertion will disrupt the expression of a trait that is potentially essential for the normal development of the plant, or can have unforeseen and undesired consequences for the plant growth and development. Even genome editing is not exempt of unwanted DNA modifications. In the case of the CRISPR genome editing, the guide RNA could tolerate a mismatch of up to 5 base pairs in the sequence, which will result in the Cas9 being activated at an off-target site in the genome. Depending on the genome editing and the type of the target sequence, up to 50% of the induced mutations could occur off-target with unknown consequences for the plant genome. In summary, conventional breeding techniques that manipulate the entire plant genome at once can result in the introduction of undesired traits in newly bred plant varieties. This happens because genes coding for undesired traits have been inadvertently included in the plant genome, because of the creation of new epistatic interactions in the hybrid phenotype, or because genes under artificial selection are pleiotropic. Genetic engineering can also result in the production of undesired genetic modifications, if it happens that the engineered genes are pleiotropic, if there is a disrupting of the gene expression because insertion of the construct in a wrong position, or if there is an off-target genome editing.